Well, hello everyone. Welcome to the NAMA Thursday night webinar. I'm Trent Blizzard and I'm excited. I'm here with Syrena Whitner. Say hi.
Hi, everybody.
Serena is going to talk to us tonight about marine fungi. I'm fortunate I saw her presentation about a month ago in Randall, Washington at our NAMA annual PNW camp. And it was very well received. There were a lot of people. They had a lot of questions. It was really a fascinating presentation on an area of mycology that we really don't often get exposed to aquatic fungi. Um, Serena lives in Hawaii. She's in Honolulu right now, enjoying the good life. She's in her fifth year PhD program, so she's getting close to defending, I guess, defending your dissertation. Is that how you put it?
Yeah, yep.
Um, and she's got a background from she's lived in Michigan, I think California, San Diego, now Hawaii, and I think based on that, Serena, I'd like to kind of bow out here, and I would encourage you to just tell us a little bit about yourself before you start talking about the fungi. I'll hop in and interrupt you as we go, so I'll apologize now for those interruptions with the questions.
Okay. Yeah, sure thing. Thank you, Trent. Yeah, I'm really excited to be here. Like Trent said, I came to the NAMA camp this year after getting a scholarship through the Mycological Society of America, working on marine fungi here in Hawaii. So it's been really good to just kind of share my love of marine fungi and just kind of introduce the community to it because they're very understudied and not a lot of people exist, myself included. Yeah, so I'm just gonna dive into it. So first of all, I always like to start off by thanking my lab group. My advisor, Anthony, is really awesome, and research just really takes a community to happen, you know. I've had the pleasure of mentoring, I think, upwards of 10 different undergrads throughout the last 5 years here in Hawaii. And I truly, truly, truly could not do any of this work without their help and their support. So they're always great. Of course, our funders such as the Moore Foundation and NOAA and NSF, and then MSA and NAMA. So everyone's awesome. It's taken a lot of people to make this research happen, so I'm really excited to share it with you guys. So just to kind of talk about myself and how I got here, you know, I think how do people study marine fungi itself is kind of a weird question because it's so random. So I grew up in Michigan down here, just south of Ann Arbor at this little red star in the middle of cornfield land called Blissfield. Here's me growing up with on a farm. So I spent a lot of time in nature, just kind of out there, you know, from our Great Lakes and just kind of walking around the fields and finding mushrooms and different animals. So I've always had this very deep connection to nature and just been like, oh, nature is so cool. And then, you know, through time that has kind of transpired into my work. So as an undergrad, I went to Michigan State University and I studied our beloved and often forgotten lichens. My first thought was, what is a lichen? Like I kind of knew what they were doing, but I didn't really know anything about them, like technically from a biological standpoint. So lichens, as I'm sure most of you are familiar, are symbionts. And they're actually a partnership between a fungi and an algae or a cyanobacteria. But what's really cool about lichens is they produce all these different chemicals or secondary metabolites. So for me, my research was focused on, you know, what fungal partners are present in different lichens, and then what secondary metabolites are produced under different environmental conditions. So is there this relationship between fungal partners and metabolites? So this is what really got me into mycology as youth. I was always into collecting mushrooms, and then I went to school and was studying ecology. And one of my professors was like, oh, I need help studying lichens. So I was like, sure, that sounds cool. Little did I know that this intro into mycology would blossom into the rest of my life. So from this point, I got really into chemistry and microbes because, you know, we're looking at these macro lichens, but there's actually a huge microbial community inside of them. So that'll transpire in a little bit, which will make more sense. But I've always had this really large curiosity of the ocean. So growing up in the Midwest, we had the lakes, which are awesome. But I never really spent a lot of time up around the ocean, but I was really into chemistry and really into environmental studies. So I just harassed a lab and Scripps Institution of Oceanography in San Diego that I just thought did really cool research. It's like, hey, this is pretty cool. You guys study chemistry. Can I have a job? And they shockingly said yes. So from there, I started working on cyanobacteria and doing natural product chemistry and looking at what these cyanobacteria are producing, under what conditions, and whether or not any of these compounds can be used for health applications. So at this point, I'm like, okay, I love mycology, but now I'm kind of working in an ocean system, but I still want to do oceanography. So how can I bridge these two gaps? So that's what led me here to the University of Hawaii. So at some point, the graduate student I worked for at Michigan State, I'm applying to grad school and I'm saying, hey, what do I want to do? I don't really know. And I know I wanted to work in oceanography. And had reached out to her for a letter of recommendation. She said, well, I know you're interested in oceanography. You're kind of tending more towards general microbes, but did you know marine fungi exist? And I said, no, that's crazy. I had no idea. And she said, you should check out this lab in Hawaii. They work on marine fungi. They're very cool. And I did not know they exist. I was like, oh, that makes sense, which I think is kind of the general consensus we all feel, say, oh, marine fungi, of course they're out there. But we don't really know what they're doing. So, that was 5 years ago. And now I am so excited to share everything that I've learned in the last 5 years about marine fungi. So kind of how I'd like to start my talks is starting very broad from just a very macro lens and then shrinking into the tiny little microbes we're going to talk about. So when we think of the ocean, I think the first thing that comes to mind is physical things. We're thinking waves, we're thinking movement, we might think of activities. Here in Hawaii, we think of surfing. I always like to joke that this is my boss. It's definitely not. But so, ocean part one. And then when we kind of get a layer deeper, we start to think of what's inside the ocean. So for most people, when you're thinking of organisms inside the ocean, the first thing that comes to mind are like large megafauna. So dolphins or corals and fish and turtles and the likes. Just kind of who's there and what they're doing. And these are what we like to call the charismatic megafauna. So these get a lot of attention, right? But what I'm most interested in are the microbes. So the ocean is kind of what I like to think of as microbial soup, where we have a huge diversity of microorganisms from bacteria to small protists and zooplankton and just things that we generally can't see, but huge diversity, and they have a lot of function in the ocean. So we're going to talk a little bit about that. But to kind of put this all into perspective, we have one single drop of seawater, or it's about 1 milliliter of water. How different abundance are these organisms? So what people might not think of first is viruses. They make up the most abundant particles in the ocean, about 10 to the 7 viruses per drop of seawater, compared to bacteria being next, which is about 10 to the 6. And then we have protists or phytoplankton, which is about 10 to the 3, which is about 1,000 now at this point. But something that we don't really have a good number on is the amount of fungi. So most estimates of fungal particles in the ocean is anywhere from 1,000 to 10,000, which when you think about it, might not seem like that big of a difference. But if we really put this into a very tactical perspective of different populations of cities, viruses are comparable to about Seoul, South Korea with 10 million people. Bacteria is about the size of San Diego with about 1.3 million people. And then the protists, which are about 1,000 per drop of seawater, is kind of similar to Randall, Washington, which is where we just recently had NAMA camp. So fungi are somewhere in the abundance between Randall, Washington, and San Diego. So when you think of it like that, that's a huge, huge difference. Like it's going from a teeny tiny little town to a giant metropolis. So we just don't really know. And then kind of the next thing I like to talk about is gearing everybody into what these microbes are actually doing with one another when we talk about energy within the ocean. So as much as I'm a mycologist, I'm also an oceanographer, and I'm thinking a lot about carbon cycling in the oceans and how these tiny microbes support the rest of the ocean. So here's the most general carbon cycle model that we have for the ocean. And I'm going to walk you through the different things that can happen. So number one, we're going to start with phytoplankton. They are the autotrophs of the ocean. They produce oxygen, they feed everybody else, and they're very, very, very important to basically every process that can occur in the ocean. So, they're fixing CO2, they're producing oxygen, and then I kind of like to think of them as bite-sized carbon. So we have these phytoplankton, the carbon is the energy source, it's the food. So when you have these energy sources, they can move and what I like to think of as 3 different avenues within the ocean. So number 1, they can go up the food chain, where phytoplankton are eaten by larger organisms such as zooplankton, which then are eaten by fish. So therefore, phytoplankton are supporting our fisheries. And from there, we can kind of go what I like to call down the food chain. So even though phytoplankton are the base, they can be broken up into smaller particles and eaten by even smaller organisms. So phytoplankton can die and turn into particulate organic matter, or POM. So they're just basically broken up into little pieces. From there, they can be broken down even further into dissolved organic nutrients. And then from there, bacteria and archaea and other very tiny little organisms can kind of munch up their different elemental pieces. So lastly, from there, we can also go out of this area entirely. So phytoplankton, when they're broken up into this particulate organic matter, some portion of it sinks and forms these giant clusters, which we like to call marine snow. So they kind of gloop together, and then these aggregates sink out of the surface of the ocean. So this is a really important process because this is our long-term carbon storage. And also I would like to highlight that all of this that I just described, minus the sinking, is happening just in the very surface of the ocean that receives sunlight. So when you think of the ocean, you're kind of thinking of everything being pretty evenly distributed. You know, we're thinking about water, so it's all very fluid, but actually most things are happening in the surface, except for things that sink out. So something that we're seeing right now is that fungi are originally missing from this model, but when we actually add them in, they're involved basically in every step of the matter. And this is very interesting because this is not a very widely accepted notion through oceanography. They're very hard to study. So people just kind of find them and then exclude them entirely because they're like, I don't really know what they're doing. But really, when we start looking at it and breaking it down, we see that fungi are involved in every single process. So marine fungi are ubiquitous in our global oceans. So here's just kind of a map of every location where we have found fungi through DNA survey. And something I do want to point out is that there's not red dots everywhere in the ocean, right? But it's not that fungi are not everywhere. It's rather that these are just the locations we have surveyed. So you can kind of see the general cruise routes that people take. You know, boats can only do so much. So this is kind of what a map would look like of that. But in reality, I need you to put into your head that every basically drop of seawater has fungi in it. So every environment we looked at, from the Arctic to the deep sea, in the middle of nowhere ocean, and basically every single organism we've surveyed has fungi. But what are marine fungi? You know, this is where I started. I said, what are they? What are they doing? And we kind of have a very loose definition of marine fungi, but basically, in order to be considered a marine organism, you need to have 3 components. So number 1, you need to be recovered consistently from water. You can't just be a one-off. You need to be able to reproduce in the water or have some portion of your life cycle reside and function in the water. And then you also need to be metabolically active in the water. So some fungi can just kind of float into the ocean as a spore, but that doesn't mean they're living in the ocean and doing anything. They're just kind of hanging out there. But as much as I would love fungi to look like this in the ocean, they don't. This is just a picture of a mushroom from a flooded riverbed in Oregon, when in reality, marine fungi are actually microscopic in situ. So it's not what you think of when you think of fungi in the ocean. A lot of people, their first question is, do we see fruiting bodies? And the answer is unfortunately no. So for the most part, things are free floating as spores, which we're seeing from all these microscopy pictures. But when you pull them out in culture and give them a solid media, they're able to grow mycelium. So very random. And then when we look at diversity just here in Hawaii, we see that they're very beautiful. And once again, there's a huge diversity. These are all samples that in my first couple of years of my PhD, we isolated around just the island that I live on and then cultured them onto agar media. So very pretty, very diverse. And a lot of them are undescribed. So the first question you guys might be having at this point beyond, what are they doing is how did this even happen? Because when I think of marine fungi, I'm thinking of something a bit on the left where we're seeing just kind of a spore hanging out, doing whatever it's doing. But I know that a lot of you are, classically mycologists, so you're more interested in large fruiting bodies. But how did we get here? So fungi actually evolved in the ocean. This is something that, once again, I was like, no way, that's really crazy. And then over time, they have lost and gained a variety of adaptations. So on the left, we have the most original version of fungi, and then moving to the right, we have evolutionary time. So all the way on the right are our most recently diverged fungi. So we're looking at things that are single spored and are just kind of floating around the water. And then right around here, fungi made a transition to land. And at this point, they lost their flagella. So basal fungi are flagellated, just kind of similar to a lot of other protists in the ocean, like dinoflagellates. They have a single spore and have a tail and locomote through the water to find either prey or food source. So when they moved to land, they no longer needed that flagella. But they also gained a lot of very important components such as hyphal growth, photoreception, a sense of gravity, and a temperature tolerance. So they adapted to this life on land, but also at some point they went back to the ocean. So how did that happen? So a lot of things that we're thinking about as marine mycologists is how a lot of fungi are actually amphibious. So here's a figure where somebody just happened to look at a bunch of different transition rates of various organisms, either from land to sea, so marine to non-marine, and then the vice versa, and then just the global rate. So this is a bunch of different eukaryotic groups. We don't need to pay too much attention to it, but basically I want to direct your attention to the fungi. So of all of the eukaryotes, fungi have the highest global transition rate of any organism, either to land, from land to sea, or vice versa. So then we look at the number of transitions. Oh, sorry about that. From the ocean to the land, we're seeing that they are about the highest. And then they also transitioned back from land back to the ocean, which is about middle of the ground. And there's really only one other group that's done that, the ciliates. So fungi have the highest habitat rate of transition among eukaryotes, like I just said. And this, a lot of people have attributed to their spore stability and dispersal. So you might remember the last slide, when they moved to land, they had all these really new adaptations because they're exposed to the elements more on land. They're having more temperature fluctuations. They're having different pH fluctuations from soil and things like that. And so they got really hardy. So when they were on land, they went back to the ocean. They said, hey, this is pretty easy. And then, yeah, so they can tolerate these high fluctuations in temperature and salinity. So they're very well equipped. They're very adaptable. So as such, many fungi are present in both marine and terrestrial ecosystems. So a lot of times when we're surveying the ocean, we find a lot of fungi that we're like, oh, this is a classic, Basidiomycete mushroom. What is it doing here? And it's actually just hanging out and living its life. So as such, marine fungi have very familiar consumer strategies to those fungi on land, including disease. So here we have a classic rust fungi infecting some plants. We're seeing a lot of fungi participating in coral disease, which is quite sad, and we'll talk about a bit more. Fungi are also parasites. So here we have our lovely Entomopteromycota or a zombie fungi infecting an ant. But we also see a lot of parasitism on marine diatoms. And then we also see a lot of decomposers. So as such, fungi are very good at decaying dead wood, just breaking it up into small components. And we also see that in the ocean. So those sinking particulate organic matter I was talking about that sinks out of the surface into the deep, fungi are known to colonize these aggregates and produce many compounds that are helping break down these aggregates into smaller components. And then what we're seeing lighting up is fungal hyphae in the particulate matter. So how do we study them? We know they're out there. We kind of have a bit of an idea of what they're doing, but how do we actually deal with these, right? So I like to talk about the marine mycologist toolkit. Number one, I work in the middle of absolute nowhere, ocean land, so we need a boat. Here in Hawaii, we have the Kila Moana. So we take this and we go about 100 kilometers offshore. And as such, we need a very enthusiastic crew. This is our safety officer, Mike. He makes sure everything's in check. And having a very good and informed team is very important. So everyone's excited. And there's kind of two different ways that I like to think about studying fungi in the open ocean. Number one is we have fungi that are associated with those sinking particles. So in order to do that, we put out what are called particle traps, and we're looking at the particle associated communities. So we have a crane that drops these little tubes like this. So the tubes are open on the top and then have a very small, tiny, tiny filter on the bottom. And we just throw these off the back of the boat with a surface buoy and we just let them go. So these particle traps are suspended in the water column at various depths, and then we're able to capture the sinking particles within the tubes at different depths. So we can say, hey, what fungi are sinking out in the surface ocean? versus what fungi actually make it to the very deep ocean. But, you know, we also know that fungi are associated with just a free-flowing water column, not necessarily particles. So to do that, we deploy Niskin bottles, very similar concepts. So we deploy these things called a CTD, or conductivity temperature depth profiler. And they have these very cool bottles. You can see they're all open right here in the bottom left picture. So these bottles, you put them out and they're all open, and then you drop this all the way down to the surface of the ocean floor. Sometimes it's about 4,000 meters. And then on the boat, we're able to control it. So we can say, hey, snap bottle B closed at 3,000 meters. And then as it's pulling up, it just keeps snapping different bottles closed. So we're able to kind of capture what's going on at different depths in the ocean. And then from there, we take this water and filter it. So looking at 2 different fungal communities, doing 2 different things theoretically. But we also want to isolate marine fungi from biological samples. So these are what we like to call as organism-associated communities. So here's a couple pictures from fieldwork we've done here in Hawaii, where we're looking for fungi in coral. And then also on the right, we're looking at fungi associated with different sponges. So each of these colors is a different sponge, and they all host their own fungal community. So it's quite fun. So, you know, now we have our biological samples, and we're kind of trying to say, okay, what are the general questions we might ask with these? So I like to think of everything kind of falling into 3 very broad categories that we'll talk about more throughout the rest of the talk. Number 1, which fungi are present in the water or any other sample that we have? What organisms do these fungi interact with? And then also, how do fungi interact with their environment? And from there, we can do a few different things to look at these samples. So I showed you some pictures from culture where you take the water or your sponge and you essentially just blend it up and stick it on an agar plate with some food, and then fungal mycelium will grow out of it. You can also look at DNA of these samples, or something that I really like to use is microscopy. So we can actually take the sample and fluorescently label our fungi, which is seen at the arrow, and then look at it under a microscope. So it's very fun. So as such, as you could probably expect, marine fungi interact with a huge diversity of organisms and their environment as well. So here's a very just simple diagram of the different places that we find fungi. And then for the rest of the talk, we're going to focus on a few different groups. So we're going to talk about fungi associated with plastic, fungi associated with phytoplankton and corals, as well as fungi and hydrothermal vents. So here's the order that we're going to talk about things. And something that I don't study, but I love very dearly, where we're going to start first, is fungi in extreme environments. So hydrothermal vents are globally distributed along the ridges of tectonic plates. They're essentially underwater volcanoes. So anywhere where you have the convergence of these plates, we're going to see a hydrothermal vent. And they're a very, very extreme place to live. So here is a picture of a hydrothermal vent. They can be anywhere between 700 and 400 degrees Fahrenheit or hotter. So very, 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 very hot. Will cook your skin off immediately if we were down there. They're very old. So they have about a 3.2 billion year relationship with microbes that we've seen from some fossil records. They also have a very high abundance of heavy metals, which, as you might imagine, influences which type of microbes and organisms can live there, and also their metabolisms. But something that you might not know is there's a large diversity of life, including fungi. You know, so what are fungi doing down here? You would never expect them to be there. But so I would also like to take a second to acknowledge how hard it is to study hydrothermal vents, because, you know, we're talking at, 4,000 to 10,000 meters depth in the ocean. So acquiring these samples is very difficult. And then pulling them back up and keeping them under similar conditions to their life in the vent in culture is also very hard. So all things considered, when we do look at the diversity of hydrothermal vent fungi, we see a lot. So on the left, we just have a picture of some cultures from hydrothermal vents versus some spores that we have isolated. And to date, there's about 50 unique fungal strains identified from these vent systems. So they're mostly dicaria, but many of them are unknown. And at this point, you might be saying, okay, well, what are they doing down there? And fungi have actually shown many, many adaptations to life on the vent, as I like to call it. So a use of complex food sources, so necromass and parasitism. Something, once again, I would like to remind you guys of is these are the ocean floor. So anything that's down there has either evolved a life down there and lives there forever, or is sinking matter that came from the surface and it's broken down into just like dead crumbles of things. So you don't really have the option to be picky about what you're eating. So you need to have a very specific metabolism to break down these food sources. But also fungi have gotten into a life of microbial competition via antibacterial production. So looking at gene transcripts of these cultures isolated from the vents, we see that fungi are producing antibiotic compounds that help fungi acquire nutrients. So not only are they acquiring these nutrients, but they're also fending off other competitors for the nutrients. And then also they have a very high tolerance to temperature and metal tolerance, as you might imagine. So it's a very unique system. Most fungi that you find at hydrothermal vents, you do not find elsewhere. And it makes for a very, very unique, like, how did these fungi get here is the question. Did they sink out of the surface? Did they evolve down there? Because, life at the surface is very different than it is at these vents. So we don't really have answers to these questions, but if anybody wants to find out, they should study it and let me know. So just summary, fungi in extreme systems, and this is only one extreme case scenario. We also have fungi in really high salt environments or very cold polar systems, but hydrothermal vents are just my favorite. So there's an unsuspected fungal diversity in these extreme environments. They have a lot of adaptations to them. They're very good food scavengers, and they also influence the greater microbial consortium. So next, we're going to talk about fungal friends and foes, as I like to call it. We're going to talk about two different case studies, both in coral and with phytoplankton. So first of all, fungal symbionts in corals. Here we have a couple SEM pictures of fungal hyphae embedding into a coral skeleton. But what are they and who are they? What are they doing? And, most people aren't thinking about fungi and coral, but I am. So first thing I kind of like to talk about with coral is the coral holobiont, or the group of microbes and other little tiny animals that make up a coral. So corals are animals, but they also form skeletons, which house all sorts of different microbes. So the most charismatic one that we like to talk about is their symbiotic algae, or the Symbiodiniae. And these are what give coral their colors, but we also have a lot of bacteria and viruses, and also a lot of random stuff we don't know about, including fungi. So when we actually take a piece of coral and we sequence the DNA, say, okay, what fungi are there? We actually see a lot of familiar taxonomy. So this is all fungi that were sequenced out of a coral here in Hawaii. So most of them are Dicaria, Ascomycetes, and Basidiomycetes, which I would also, one thing that I haven't yet described that I would like to point out is a lot of things are related to database bias. So we're seeing a high abundance of these taxa in coral, and that's not necessarily because other things are not there, but we just don't have very good representation in a database because they're very cryptic, they're undescribed, you don't really know a lot about them, so it's very hard to tag them. But in general, these are things that we're seeing in coral. But we also have some novel diversity that's not described whatsoever. So when we're thinking about friends and foe, fungi have a lot of very beneficial interactions with coral. So number one, they can increase nutrient cycling through different metabolism. They also provide thermal protection by producing various compounds and kind of like a fungal sunscreen, as we like to call it. And they also can protect from these harmful microbes. So the same way that fungi are producing these antibacterial compounds in vents, they also can produce antibacterial compounds to fend off bad bacteria. They also can produce antifungal compounds and just kind of basically prevent nuisance microbes from taking over coral. But fungi can also be very, very harmful. So these are the fungal foes. So on the left, we're seeing a very healthy Gorgonian coral. He's having a good time. He's existing. His microbiome is happy and normal. But then, let's start to increase some type of stress. So maybe that's ocean temperature or excess nutrients from coastal runoff. And our corals, they start to get stressed. And when you have stress, you have physiological stress to also the microbes that inhabit them. So corals themselves are very resilient, but their microbes might not necessarily be. So when we add all of these stressors in, we can see something called Aspergillus disease, where these Gorgonian corals are taken over by Aspergillus sedoi and form just these degenerative pockets where the corals are basically broken down by this nuisance fungi. And at this point, This spreads very easily because we have a lot of stress within the coral community in general. So these fungi are able to colonize the adjacent corals and just kind of break everything down. And whether or not the fungi is able to recover to its original state is something that's very up for debate, and it's very dependent on the disease. These incidents of stress that cause various diseases are usually sporadic, right? So we're very familiar with coral bleaching, I'm sure, where we have these periods of high temperature and the corals get stressed and expel their algae, and then they're taken over by these nuisance microbes. And at some point, the temperature does decrease and go back to normal after a heat wave. But whether or not the original microbes are able to outcompete the now nuisance bad microbes is something that doesn't always happen. So we're seeing like large die-offs in coral, which is very sad. And this is only one of about 5 diseases in coral that's caused by fungi. So very problematic. We don't really have a solution for it yet, but so fungi can be very helpful or they can be very bad. And kind of going back to the microbial world, fungi are very important phytoplankton pathogens. So phytoplankton, like I said, are the base of the food web. So here's a picture of a diatom, which is kind of this bar that we're seeing, but also the yellow arrow is pointing to a fungi from the phylum Chytridiomycota. So these are very old, very basal fungi that evolved in the ocean and parasitize all sorts of things. And something I really want to kind of hone in before we get too far is phytoplankton produce 30 to 50% of our global oxygen. So here's just a satellite image of chlorophyll bloom. So chlorophyll is equated to fungal or phytoplankton abundance. So overseas, and we're seeing these huge blooms from space. So every third breath you take, you should think of phytoplankton. So not only do they support fisheries and every process in the ocean, but they also produce all the oxygen we breathe. You know, I think a lot of people attribute our oxygen to the rainforest, but really you should be thinking about phytoplankton as well. So we have our phytoplankton that are very important to us and to the ocean. And then we also know that they're parasitized by these chytrid fungi. So before we get too deep into talking about that dynamic, I really like to talk about chytrid fungi life cycle because they're very typical fungi that most people aren't thinking about, and they're very different, right? So starting up at number 1, we have our aquatic zoospore, which is a single spore with our flagella. So our little spore, he's swimming around and he's finding a host, usually via chemotaxis. So our phytoplankton are producing these chemicals, they're dissolving into the water column, and fungi can you can kind of think of it as smelling, where they sense these chemicals and they say, oh, I want to go over here, here's a host. So going to number 2, our zoospore will embed into the host and develop into a thallus or a fruiting body. So our little flagellum or its tail will embed directly into the host, kind of like a straw, and from there it will grow a network of rhizoids, so kind of like a bunch of roots. So we have this attached thing with a bunch of anchors. And then from there, we go to step 3. This thallus will mature and form a bunch of different zoospores inside of it. So one single thallus can exponentially grow into thousands of new zoospores inside of it. And at some point, moving to step 4, it just gets too big and there's too many zoospores inside of it, and it just pops open. There's this little hole at the top you can see. And all of these zoospores then enter the water column. So we're talking exponential growth here. So you can see how if there was an infection, oh, sorry, this keeps happening. If there was an infectious cycle, you could see how that could spread very quickly to the surrounding area, because you're starting with just one infection, but one infection turns into 100 new zoospores. So when you have more zoospores, you just have a higher rate of encounter. So this can spread very quickly. And when we're talking about this in the dynamic of a phytoplankton bloom, they can actually have huge, huge pressures on phytoplankton bloom termination. So here is a figure. We don't have to go too much into it, but I'll explain what's most pertinent. So the straight lines are phytoplankton cells per liter, or just cell density. How many cells do we have? And then the circles are percent of the population infected. So blue is how many cells we have. So let's say we have about 1,000 cells at this time point. And then we have this many phytoplankton infecting them. So that's about 80% of the phytoplankton are infected. And when they're infected, they're usually being killed. They're sucking out all of the nutrients. So then right after we see this high incidence of infection, we see a phytoplankton bloom crash. So this line goes all the way down. And then there's no more food for the chytrids. So then the chytrids kind of die off. But then that cycle essentially repeats itself where, okay, we have no parasites, our phytoplankton can kind of increase, and then boom, huge infection, and then it drops off again. So this is what we call a boom and a bust cycle. So something that I'm very curious about and I work on specifically is how climate change will impact these relationships. So why does this matter? And excuse me, back to the concept of stress inducing different host switching that we saw in the corals. So here we have 4 different phytoplankton by the little cartoons, and each one has its own chytrid fungi attached to it. So under normal, happy, well, not happy, but under normal ambient conditions, we might see some very specific host associations where specific chytrids only really infect specific phytoplankton. But then you include climate change, so all these different physiological stresses, and it's stressing out the host is what we need to be thinking about. So the host gets very stressed, they kind of put down their defenses, and then all of a sudden we see host switching. So these chytrids might be able to infect different hosts or infect multiple hosts, and we're seeing things that all of a sudden are less resilient to parasitism under climate change stress. So we're already seeing this in a case study that some of you might be familiar with the chytrid fungi essentially wiping out thousands and thousands of frogs and other amphibians across the world. So this chytrid fungi, I'm not going to say it because I'm going to butcher it, but we call it BD chytrid. These frogs have been stressed by temperature and they're more susceptible to the chytrids. And essentially once they're infected, it alters their mucous membrane. So amphibians need that mucus coating in order to be able to breathe. And it closes up their pores, their mucus, and so all the frogs and all the amphibians are suffocating. We're having a mass wipeout of many, many species are just going extinct. So it's really sad and it's very problematic. So something that I wanted to talk about that's not super far off, but is obviously a dramatization. So the show The Last of Us really talked about this, where we have these stresses and all of a sudden people are now susceptible to these fungal parasites. So obviously it's very dramatic, but it's not too far off because it's happening in our frogs. But kind of taking it back to the phytoplankton, we're also seeing very similar things happening under climate change scenarios. So on the left, we're looking at a graph of phytoplankton abundance, and on the right, we're looking at a rate of chytrid fungal infection. So I'm going to go through this a little bit more in depth. So the closed-in dots are going to be a warming treatment. So they ran an experiment where they had 2 different tanks. One was natural water, and then one was increased temperature, and then they just compared between the 2. So at the yellow arrow, we're looking at the warming treatments for phytoplankton. And then on the right, we're looking at timing of chytrid infection. So if we look along the bottom, axis, we're looking at time. So under these warm treatments, we're seeing lower phytoplankton cell densities, and that might be due to stress where they're just not able to reach these high densities that they usually would without the stressors. But when we look at chytrid infection, which is often left out in most oceanography studies, we're seeing a totally different picture where it might not be necessarily that these phytoplankton aren't able to survive, but rather they're being wiped out at a sooner time point. So compared to control or the open circles, we're seeing that chytrid infection is actually about the same proportion. So we're seeing the same number of cells infected, but it's just occurring sooner. So if we have more cells being wiped out earlier, we're just not able to reach these peak densities. So putting this all together, if we have higher rates of infectivity, we're going to have less phytoplankton, which is bad because that essentially means less food for higher trophic levels like zooplankton and fish, but also less oxygen for us. So, you know, this is about 25% less phytoplankton under a normal case scenario. So it's pretty significant. And that's looking at just warming. This isn't including nutrients or pH or any other stress. So it's a bit scary, but I like to think that microbes are resilient, so maybe it won't be the case. But so just kind of summarizing the fungal friends and foes, when we're talking about coral-associated fungi, they can help or hinder coral health depending on the fungi or the environmental stress. They can also impact reef stability. So about 1/3 of marine life spends some portion of their life on a coral reef and rely on other food sources that are found there. So when our coral reefs are dying, we're losing that reef habitat that lots of fish use. So that impacts fish availability for food and, you know, the whole lot of things. But they can also alter the coral microbiome specifically. And then when we talk about phytoplankton associated fungi, they have huge impacts on phytoplankton bloom termination. So when they're infecting more fungi earlier on, we're having less phytoplankton available. And therefore that has an impact on oxygen production and also the food available to the higher trophic levels. So last thing we're going to talk about is an experiment that we did here in Hawaii of fungi versus plastic. So plastic is the largest new habitat on Earth. We're all thinking about it. We're all trying to use less plastic, and it's a huge concern because we know that plastic does not break down well. So as of 2010, this is very outdated. There's about 8 million metric tons. I think that's about 50 now, last time I read. And there's a rising plastic production still to this day. And this has all sorts of just bad implications for marine life, including entanglement, ingestion, and then toxicity. So I think some statistic is we eat about a credit card worth of plastic a year through fish and other things. So it's really nasty, and these chemicals can reside in us forever. But something else that we're thinking about is these microplastics, right? So they're breaking down these smaller components that we are eating, that fish are eating, and it's just very bad for us. But as such, plastics have become a huge target for bioremediation efforts. So we decided to see if fungi could do anything about the plastic problem. So as such, we went around Oahu. Here's our island, and we collected fungi, or different samples, I should say, to isolate fungi from around the island, just basically anything that we could grab. We collected from water to sediment to sponges and corals. And then from there, we isolated about 450 unique fungi. So here's what we like to call the bucket of fun. It was very horrifying, just a bucket of a bunch of isolates going crazy. And then from there, we wanted to inoculate this plastic media with marine fungi to see if they could break down these plastics. So this project was mostly fielded by my old undergrad, Rania Steinbeck, who is an absolute superstar, one of the most brilliant people I know. And we use plastic degradation assays using high molecular weight polyurethane and polyethylene additives. So essentially what we did is we have this the general agar plate, but we replaced all of the other carbon source and all the other food that we usually use with these plastic additives. So basically, if the fungi wanted to survive, the only thing they could eat was plastic. So how do we do this in the lab? We're measuring based on degradation or rate of clearance. So here we have two fungal strains, strain A on the left and strain B on the right. And they're inoculated with these little tiny, they're kind of like a hole punch. Like you take a fungi and you just kind of stick it on there. So these little circles are our fungi. And let's say that strain A and strain B are inoculated for the same amount of time, which is 1 week. So how we measure the clearance rate is the zone of clearance. So essentially this little halo around our fungal isolate. So strain A has a larger zone of clearance compared to zone B, or strain B. So if we're measuring based on the same time, we would say that strain A is a faster degrader compared to strain B. So we have all these isolates. We decided to test them, say, okay, who can eat plastic and who can't? So we tested about 200 of the isolates that we had. And of those 200, about 62% of isolates were able to degrade plastic. So here we have a figure of the different fungal genera that we tested. So on the x-axis, we have our fungi. And on the y-axis, we just have number of isolates tested versus whether or not they were successful. So for example, all the way on the left for the penicillus or penicillium fungi, we tested 15 isolates, and of those 15, 9 of them were able to degrade plastic. Very similarly for aspergillus, where we tested 16 and 12 were able to degrade plastic, and so on. So it's a pretty high proportion. Granted, I want you guys to think about the fact that the only thing that they can eat is this plastic if they want to survive. But it just kind of speaks to how adaptable fungi are. So when we're looking at the clearance rates of different fungi specifically, so different fungi do different things, and everybody's built different. So some fungi were able to, if we're looking at the orange line, you know, he took off very, very quickly. And by day 12, he had basically eaten everything and was then just kind of hanging out. Versus some fungi, like this dark blue or the aspergillus, did not clear anything for 42 days and then all of a sudden cleared 90%. So different fungi might be better suited for these bioremediation efforts because they're able to grow faster, versus a lot we're just kind of middle of the line. So then from there, we said, hey, these fungi are, some of them are faster, some of them are slower. So that kind of led us to believe that they have these different genes they're producing that might help them be faster. So we said, okay, well, can we evolve them to degrade more plastic? So basically then what we did is we would scrape off some of this mycelium and transplant it into a new media every, I think, about a week or so for 6 months. And every time we would transfer them, we would put a higher concentration of this plastic. So we were essentially force feeding them these plastic compounds. And if they wanted to survive, they were just going to have to eat it. And we look at that as data, we then decided to race our quote, quote, evolved strains against our original strains and compare that clearance rate that I showed you in the last figure. And some of them were able to do it faster. So some of them grew faster, some of them died entirely. They did not like the plastic, and some of them were the same. So in total, about a third of the strains were able to degrade this plastic faster with a rate increase of about 11 to 20%. So this is pretty substantial. And I would also like to point out, we're not using any high technology. We're basically just adding some plastic compounds to some media and letting it grow. We're doing no sort of like gene modification, and we're not looking at what genes are involved in breaking down this plastic, but we just know that they can do it. So they're essentially very good targets for it. So in conclusion, fungi might be a very promising bioremediation effort. These fungi are naturally found in the ocean and can naturally degrade these plastic compounds. Some strains can do it faster, and therefore they might be a very good target for these efforts. But something that I do want to kind of hone in on is that, you know, we're feeding them this plastic and they're able to eat it, but in a naturally occurring environment, they have other carbon and other food sources available to them. So do they prefer these carbon sources? Because naturally, I think fungi are going to want to eat what they usually eat and not necessarily plastic. So kind of working on that issue and maybe modifying fungi to only like plastic if we know how they're doing it, or just deploying a bunch of fungal cultures that we know degrade plastic in the event of an oil spill or into a garbage patch, and seeing if they take advantage of that is kind of one question or the other. But so just kind of to wrap everything up for the end of the talk, marine fungi are dynamic and ubiquitous in marine ecosystems. They're found everywhere. And quite literally everything that we've looked for has found fungi on it. They interact with various organisms in their environment, and they're doing all sorts of different things. And they also have a huge influence on these global biogeochemical cycles. So when we're thinking about the phytoplankton, you know, they're decreasing oxygen production, they're increasing particulate matter as they break them down. But in general, marine fungi are still a huge black box. I am one of like 45 people in the entire world that study marine fungi, compared to the tens of thousands of people that study any one specific thing. So it's an incredibly exciting, but also very challenging field. And with that, I'll wrap this up and take any questions. Thank you, everybody. And then I see some questions in the chat, so I will go there first. Okay, so the first question I see in the chat is from Sam Weiss. And he said, are the metabolites from eating plastic a concern? That is a really good question. I would offhand say no. Most metabolites tend to be pretty specific and are only produced under certain conditions. So if they're eating one or the other, they're probably only using those metabolites under these case scenarios, is what I would say. So I wouldn't think it's a concern, but we're also not entirely sure, because unless you're testing these metabolites under different treatments in the lab, there's no real way to say. So with a healthy dose of skepticism, I'd say it's probably okay, but not really entirely sure. And then, so Trent, should I stop sharing my screen and then we can all just kind of talk? Or what do you think is the best avenue?
I think we got a few more. Do we have a few more questions in there? Um,
I don't see any more questions in the chat.
we have one from Ashley just came in.
Oh yeah. Are there known freshwater fungi currently being studied? Yeah, okay. So most of the information on marine fungi we're actually basing off freshwater fungi. So yeah, in short, yes, there are a lot of aquatic fungi that are studied, especially chytrids. They infect phytoplankton also in freshwater lakes, and chytrids that infect these frogs are found in ponds and also in soil. So I think those are the most abundantly studied fungi in aquatic freshwater ecosystems. But there's also a lot of information on pollen degrading fungi in ponds and lakes and stuff like that. So in short, yes. It's also much easier to study them, just in the sense that sample collection is very easy comparatively. You can just kind of go out on a small boat into most lakes and get some water. Whereas, for us, we have to go out on a research cruise and go out into the middle of nowhere ocean and drop this really expensive equipment. So the trade-offs are in just sampling are very evident in the research available. So yes. So Donna says, are there any edible aquatic fungi aside from we might consume eating some ocean foods? I don't know. I do not think so. I think that excuse me, I'm a little sick. So most fungi in the ocean are not forming fruiting bodies, and they're just as spores. But something that I would be curious of is, so they're existing as spores in the ocean, but then we take them in the lab and we can culture them. So one of the classic fungi that I see as spores in the ocean is like Agarica fungi or Tremella fungi. I see a lot of those in my data. But then when I take them in the lab, they're mostly forming mycelia. I haven't seen any fruiting bodies yet. So it's possible that if you gave them a food source, we could grow fruiting bodies, but I don't think anybody's ever done that, to my knowledge. But there are spores of aquatic fungi in the ocean for sure. Can some of them be interchangeable between fresh and salt water, says Ashley? Yes, super good question. So a lot of fungi are found in both. And one of the really most, of all of the marine fungi people in the world, most people are studying coastal marine fungi. So one of the huge research locations is in mangroves. So these are estuarine environments that are partially fresh and partially salt, and they fluctuate based on different tidal conditions and seasonal variation. And we're seeing fungi that can exist in both, and they have these really, really, really high salt tolerances and usually associate with mangroves or something like that. But in general, yes, we do see fungi in both. That's a fun question. Ooh, Scott, I love your question. So Scott Clark says, are you using flow cytometry to isolate marine fungi? And yes, that is something that I am currently working on. It's very, very challenging, but we're doing it. So for those who are not aware, flow cytometry is a method that people use to essentially count or isolate microbes. So you can take the tiniest micro drop of water or some solution, whatever you put them in, and it goes through a tube and it will flash light at them so you can see if you've like fluorescently labeled anything. So for fungi, we will add a fluorescent dye to our samples that binds to chitin in their cell walls. And when you flash a light at it, it'll say, okay, anything that fluoresces with this chitin stain is probably a fungi. And then from there, we can kind of pick them apart and isolate them into different little teeny tiny tubes. And then we can sequence them. So yes, we do that. It's a bit challenging because fungi are mostly spores. So it's pretty easy because they're all pretty small. But some fungi are filamentous in the ocean, and they can be like 3 orders of magnitude larger. So it's a bit challenging for flow cytometry. Also, when fungi, I've tried to use flow cytometry to isolate fungi that are parasitizing phytoplankton. So when you have two things attached to each other, sometimes it can separate them out and sometimes it can't. But then if it does not, it's able to say, hey, this is the phytoplankton's attacking. But it's not routinely used, unfortunately, between marine mycologists. That's something I really push for. I think something, if I could just go on a quick tangent about being a marine mycologist in a very, very understudied field, in a trying to find some navigational path in the large, massively studied world of oceanography, is most people that are studying marine fungi are either people who have a classic mycology background. So my boss is a very good example of this, where he studied mycology and he studied shiitake and maitake for his thesis, but has gotten interested in oceanography and marine fungi. But he's applying classic mycology methods, where there are the totally opposite end of the spectrum of the oceanographers who are trying to apply just oceanography-based methods to it. So there's a bit of a disconnect between how we're studying marine fungi, where we're either mostly treating things like general mycology when we should be probably thinking more about oceanography methods. So in short, yes, Scott, we are. But it's also like no methods are developed for it. So it's like the Wild West. Everything I do, I will go online and try and find some paper that's done it before. And I'm like, oh, well, that does not exist. I guess I have to play with it for 6 months and see what happens. So yeah, it's very frightening, but it's fun. So Brooke has a question. So are you seeing any practical applications for what you do specifically, or is studying marine fungi more basic science, data gathering focus? Yes. Okay. So Brooke, I totally know what you mean. So yes, it's a bit of both. So one of the things that I work on is specifically with the phytoplankton parasites that I did not talk about in this talk, but is more of a practical application. So I work a lot with the fisheries section of NOAA here in Hawaii. So there's been some really interesting studies recently about phytoplankton cell size correlating with catch rate of different fish. So for us, we're mostly looking at modeling for tuna. So the average phytoplankton cell size of the community, if it's larger, is going to result in a higher tuna catch rate about 5 years out. So that's basically, we have, oh, I wish I had a slide for this. I do in a different PowerPoint, but the general gist is that the larger the phytoplankton, the more fish we're going to have, because it's a better food source for the zooplankton, which is a better food source for the juvenile fish. So when we have more plankton and larger plankton, we have more fish. But so what I work on specifically is under climate change scenarios, so ocean warming and ocean acidification, do we see a difference in the different cell sizes that the fungi parasitize? So are fungi essentially dropping the average phytoplankton size? Because most parasites want a better bang for your buck, so you're going to eat a larger plankton if you have the option, right? So if fungi are primarily eating all these large phytoplankton and only leaving the small ones available to go up the trophic food web, we can put that into modeling data and say, hey, under these different climate change scenarios, we're going to see a smaller average phytoplankton cell size. So we're going to have a smaller tuna catch. So that's actually the main basis of my thesis, is modeling parasitism as it relates to fisheries success. So yes, but also there is a huge component of just general discovery. So we really don't know what's out there. So a lot of my work is just generally like going around and collecting fungi and describing them and saying, hey, look at all these fungi. But once we do that, we can kind of pave the way for different, more interesting questions. So how do fungi impact the amount of carbon that sinks is a big question that a lot of people work on with bacteria. So that's very important for like long-term carbon storage and just removing carbon from the atmosphere. So people care a lot about that. But yeah, there is some practicality. There definitely is a lot of very basic, like what's going on out here type stuff. Yeah. But so yeah. Thank you, Brooke. Okay, we have a question from Jared. If money weren't an object, is it realistic yet to actually apply known plastic degraders to something at a large scale, like a plastic garbage patch? And would that just be released in the form of mix of liquid cultures. Okay, yes. So this is totally something that I think would be awesome. And if money were not an object, I think that we could do that. So there's a lot of studies on on oil spills actually, where right after an oil spill, fungi are usually the first colonizers of this oil. So a lot of fungi are just naturally degrading oil and plastic laden compounds. But something that I think would be really, really, really cool is, like I said, these like we're force feeding them plastic compounds, right? But I think if they had the choice of an easier to break down food source, because plastic compounds are really hard to degrade. So, you know, naturally I tend to think they would like things like dead matter instead. I think if we could genetically modify fungi that we know degrade fungi, and we could kind of see how they do it, or they degrade plastic, sorry. If we know how they're degrading the plastic and we can kind of modify them to only like to eat plastic, I do think that it would be a pretty cool type of thing to put fungi out into the ocean and let them break down large amounts of plastic. And I would suspect that we would probably take, so what we can do is we can take cultures that are growing on agar, scoop them up and put them back in water and just resuspend them as spores. And so I would probably put a spore slurry out there personally. What I would do. All right. Next question, Donna. What are the products of, so Donna says, what are the, I'm actually not sure if you guys are reading the chat, so I'm going ahead and reading them also. What are the products of plastic or oil consumption mediation by fungi? Are toxic? Yeah, okay. So this is a really good question, Donna. So we know that fungi are breaking down plastic and they do it, but we have no idea what the byproducts of this is. So plastic compounds in general are like very, very, very intense hydrocarbon structures. So they're hard to break down, number one, but also breaking down hydrocarbons usually releases a lot of energy and also produces, yeah, a lot of byproducts. So in short, we have absolutely no idea. I think it probably depends on the plastic source more than anything, what the byproducts would be from that. Yeah, that's definitely a very high potential / we do not know. Yes.
Well, thanks, Serena. I think we're down to our last question there.
Yeah, I think that was the last question. If anybody wants to not be shy and say it out loud,
I'd
also
like
welcome.
to ask a question, which I didn't type in. You know, you talked a little bit about it. Do you have a recommended path for a young person interested in becoming a marine mycologist?
Yeah, totally. My like number one thing that I would say is like just being super curious and like building good random skill sets. So for me, like I had worked on fungi and I had done some oceanography work, but I had no actual experience in marine fungi whatsoever when I started school. And I basically just, you know, emailed my boss, my now boss, was like, hey, this is really awesome. And I have some relative experience in some adjacent type fields, like, and I have a really good attitude. You should give me a job. And he was like, sounds good. So I think like just asking really good questions and being really enthusiastic, number one. I think having like something that blew me away that I really want to emphasize here is at NAMA, like how much people who like, because I come from a very like academic training, right? But how much people really know about mycology. Like everybody loves fungi. So I think just having any background and any knowledge in fungi, like I was blown away. I was like, oh my God, you guys love fungi. This is awesome. You know so much. Probably like more than I do about a lot of especially terrestrial stuff. Um yeah, just like being really enthusiastic, reaching out to people that you know do interesting work and trying to get some type of leg in the door. I'm a big fan of just like emailing until you get a response and just being really stoked. yeah, local landowners, nature centers, conservation groups is great. Um, to date, I don't know any sort of conservation groups involved in marine fungi because we just don't have like enough information to say, hey, how are populations doing, which ones are important, and are they facing any threats to our knowledge? We have none of that, which is a bit of a bummer. Um, but yeah, making friends in random places is always the best. Just talking to people.
Awesome. Thank you.
Yeah, of course.
All right. Well, I guess it's a good time to thank you officially, and we're grateful that you came to our foray, and that you also
Yes.
agreed to do an online talk for everybody that couldn't make it to that. So
Yes.
thanks so much for really being involved with NAMA.
Yeah, I'm so happy to be here. You guys are an awesome group, and I love spreading the word of the marine fungi. I think they're really fun. And if anybody also has any questions, like please email me to kind of like follow up on what Trent said. If anybody wants to actually get involved in marine fungi research, we're looking for students in our lab currently. We have a really fun group, and living in Hawaii and studying marine fungi is awesome. So if you know anybody or you are yourself, like please reach out. And I just love like talking stories. So if you ever just want to pick my brain, please call me or text me or email me. Any of the above.
Thanks. You're getting a lot of very positive comments in the chat. So it looks like people
Yes.
really.
Thank you so much, everybody.
All right. Good night.
Bye, guys.